The human body is constantly at work. It is never completely at rest. For example, even when we are sleeping, our lungs are busy inhaling oxygen and exhaling carbon dioxide. Carbon dioxide is a waste product generated by our body. Like carbon dioxide, various other waste products such as ammonia, urea, uric acid, water, and excess ions like phosphate, sulfate, sodium, potassium, and chloride are produced in our body as a result of different metabolic activities like ingestion, digestion, and respiration. These waste or excretory products, if retained in the body, may prove fatal and so must be totally or partially removed from our body. The process of elimination of metabolic waste products from the animal body is called excretion. Among the different waste products, ammonia, urea, and uric acid are known as nitrogenous wastes. Of these, ammonia is the most toxic and requires a lot of water for its elimination, and is thus excreted only by those organisms that can compensate the loss of water. Generally, bony fishes, aquatic amphibians, and aquatic insects excrete ammonia in the form of readily soluble ammonium ions through their gills or body surface by the process of diffusion. Animals that excrete ammonia are called ammonotelic, while the process is called ammonotelism. As excretion of ammonia requires a lot of water, terrestrial animals have evolved a mechanism to convert ammonia into less toxic forms of urea and uric acid, which help conserve water. Mammals, including humans, and many marine fish and terrestrial amphibians excrete urea. Excretion of nitrogenous wastes in the form of urea is called ureotelism, and such animals are called ureotelic animals. In these animals, the liver converts ammonia into urea by the ornithine cycle and releases it into the blood. The urea in the blood is then filtered by the kidneys and excreted. However, in some animals, a small amount of urea is retained in the matrix of the kidney to maintain the required osmolarity. The least toxic form of nitrogenous waste is uric acid. Though excretion of uric acid involves a lot of energy, it helps conserve water considerably, as it is excreted in the form of pellets or paste involving very little water. Some animals excreting uric acid are reptiles, insects, land snails, and birds. Such animals that excrete nitrogenous waste in the form of uric acid are called uricotelic animals, and the process is called ureotelism. Did you know that in mammals, uric acid is formed by the degradation of nucleic acids and is excreted as a part of urine? As animals excrete nitrogenous wastes in different forms, the structure of their excretory system also varies. The study of phylogeny of the excretory system reveals that primitive animals such as invertebrates possessed simple organs like protonephridia or flame cells, which in the course of time evolved into complex organs such as the kidneys in vertebrates. Flame cells or protonephridia are the excretory structures in animals like platyhelminthes, rotifers, cephalochordates, and certain annelids. Apart from performing the function of excretion, these cells help in osmoregulation, or the regulation of the fluid volume and ionic balance in the body. Another excretory structure is the nephridia, found in many annelids like the earthworm. Nephridia are tubular structures that help remove nitrogenous waste and maintain the fluid and ionic balance. Malpighian tubules are excretory structures found in arthropodans such as insects and arachnids. These tubules, along with removing nitrogenous wastes, perform the function of osmoregulation. The antennal gland or green gland is the excretory structure in crustaceans such as the crab and crayfish. This gland performs the function of excretion and osmoregulation. The kidneys perform the main function of excretion in highly evolved mammals such as man. Along with the kidneys, the skin, liver and lungs also help in the removal of certain wastes from our body. Our skin has sebaceous glands that secrete sebum, through which substances like hydrocarbons, sterols and waxes are excreted. This secretion, however, provides a protective oily covering for the body. Like the sebaceous glands, the sweat glands on our skin also help in the removal of excretory wastes. These glands produce sweat, a watery fluid containing sodium chloride and small amounts of substances such as urea and lactic acid. Moreover, sweat also helps to cool the body's surface. Another organ that plays an important role in excretion is the liver, the largest gland in our body. It breaks down drugs and reduces their toxicity by drug metabolism. It also detoxifies ammonia and converts it into urea, which is eliminated by the kidneys. Moreover, it degrades hemoglobin, hormones, drugs and vitamins and adds it to bile as bile pigments bilirubin and biliverdin, which are then excreted through the feces. The lungs are also excretory organs that help in the removal of large amounts of carbon dioxide, a byproduct of cellular respiration, from our body. In addition, the lungs also help in the removal of water. Besides the kidneys, the skin, liver and lungs, saliva too helps in the removal of small amounts of nitrogenous wastes from our body. Therefore, excretion is an important process which is vital for all living organisms. The excretory organelles may differ from animal to animal and have evolved from simple to complex structures based on the needs of the organism. The excretory system is a biological system that helps in the removal of metabolic waste products from an organism. Much of the excretion in man is executed by the urinary system, which constitutes a pair of kidneys, a pair of ureters, a urinary bladder, and the urethra. Of these, the kidneys play a very vital role. They filter blood by removing nitrogenous wastes, regulate blood pressure, and assist in the production of red blood cells. The kidneys are compact, bean-shaped organs on either side of the spinal cord in the lower back. More precisely, they are situated between the last thoracic and third lumbar vertebrae in close proximity to the dorsal inner wall of the abdominal cavity. The 11th and 12th ribs partially protect the upper part of the kidneys. The right kidney, which is near the liver, is slightly lower than the left one, which is adjacent to the spleen. The kidneys are reddish-brown in color. In an adult, each kidney measures about 10 to 12 centimeters in length, 5 to 7 centimeters in breadth, and 2 to 3 centimeters in thickness. Each kidney weighs about 120 to 170 grams. The kidneys have an outer convex surface and an indentation or a notch on the inner concave surface called the hilum, through which blood vessels, nerves, and the ureters pass. The kidney has a tough, fibrous outer layer called a capsule, and its longitudinal section shows that its inside is differentiated into two main regions, an outer cortex and an inner medulla. There are about 8 to 18 cone-shaped structures in the medulla called medullary pyramids, which project into the calyces. The tip of each medullary pyramid is called a papilla. The cortex protrudes or extends between the pyramids as renal columns called the columns of Bertini. The medullary pyramids project into the calyces, which lead to a wide funnel-shaped space called the renal pelvis. Each kidney contains about 1 million nephrons in the cortical region, which are the basic functional units of the kidney. The nephrons filter nitrogenous wastes and excess salts from the blood and help in body fluid regulation and urine formation. Each nephron has two parts, the glomerulus and the renal tubule. The glomerulus is a ball-shaped network of capillaries formed by the afferent arteriole, which is a fine branch of the renal artery. It performs the first step in filtering blood. The filtered blood from the glomerulus exits through the efferent arteriole. The other part of the nephron, the renal tubule, helps in the reabsorption of salts and water as the urine passes through it, thereby concentrating the urine. The renal tubule begins with the Bowman's capsule and consists of proximal convoluted tubule PCT, Henle's loop, and distal convoluted tubule or DCT. The Bowman's capsule is a double-walled funnel-shaped structure that encloses the glomerulus. It collects the glomerular filtrate. The Bowman's capsule, together with the glomerulus, is called the Malpighian body or renal corpuscle. The Bowman's capsule opens into the proximal convoluted tubule, a highly coiled structure, and is followed by a hairpin-shaped loop called Henle's loop. Henle's loop has a descending limb and an ascending limb. The ascending limb opens into the distal convoluted tubule, which is also a highly coiled tubular structure. The distal convoluted tubules of several nephrons open into a collecting duct. Several collecting ducts converge and pass through the medulla into the renal pelvis, which leads to the ureter. The collecting ducts, along with Henle's loop, lie in the medulla, while the Malpighian body, proximal convoluted tubule, and distal convoluted tubule of a nephron lie in the cortex of the kidney. The nephrons in the kidney are also distinguished as cortical nephrons and juxtamedullary nephrons based on the length of Henle's loop. Cortical nephrons constitute the majority of nephrons and have a very short Henle's loop that just about extends into the medulla, while the juxtamedullary nephrons have a very long Henle's loop that runs deep into the medulla. Each nephron also has a fine network of capillaries called peritubular capillaries, which is formed from the efferent arteriole leaving the glomerulus. A minute vessel from this network of capillaries forms a U-shaped structure called the vasa recta, which lies parallel to the loop of Henle.